Hi everyone, thanks for coming. Welcome to this event and for International Women in Engineering Day 2021. My name's Solace. I'm a third year, well I just finished my third year of engineering at Oxford at St Peter's College. And so today we're going to have an event which will be a short talk followed by a Q&A session. So feel free to put any questions into the Q&A box and we'll hopefully get through as many as we can. If anyone has any trouble, please email the email that's on the bottom right of the screen just there. And the event will be recorded in case you miss anything, and live captions are available if you need them. So today we have Professor Cathy Ye, who is an Associate Professor of Engineering Science, Director of the Oxford Centre for Tissue Engineering and Bioprocessing, and a Fellow of Linacre College, and she's going to be speaking about stem cells and engineering. What is the link? So yeah, Cathy, thank you.
Thank you, Solace, for the introduction. Am I muted or am I okay?
No, I can hear you.
Yeah, because it says unmute your mic, so I'm fine. Okay, that's. Thank you. I'm going to share my screen. Is it playing? Sorry, I'm just trying to get it into full screen mode. Yeah, can you see it's playing in full screen mode?
Yeah, I can see that.
Okay, that's good. Sorry. Good afternoon, everybody. So you can see this picture here. Why I'm putting it here is because I was given this T-shirt a few years ago on the Women in Engineering Day, like today, and so I quite like it. And today I'm wearing it. Because you are not able to see it, that's why I'm putting the picture here to show I'm wearing this T-shirt today. Okay, so yeah, as Solace introduced, that I work in this department in the Institute of Biomedical Engineering, so that's my specialty. As you may know, in this department, we have general engineering, so we have people working in different branches of engineering, and I work in biomedical engineering, but I will give you more background, my history. Okay, so, why it's not moving? Okay, so a bit of background about myself. So I was born in China. So I was educated in China and I didn't have any chance to visit other countries until I came to study in Oxford in year 2000. So I studied chemical engineering in university because I quite like chemistry in my high school. And when I arrived here, or when I, finishing my undergraduate, I was considering doing PhD, but in which field? So at that time, I was looking at, different things I can do as a chemical engineer. And then at that time, the concept of tissue engineering just came around. So I was fascinated about this field. So I will talk about it later. But so I arrived here in a new country. I need to speak a new language and I need to learn biology, as part of, this tissue engineering subject. So yeah, it was very tough at the start because, for biology, I learned it in my, high school. It was in Chinese, so I never learned biology in English. So yeah, so it was a tough start, but I survived. And then, so I continued my research in this area in tissue engineering from my PhD time So after my PhD, I went to work in Imperial College, London, as a postdoctoral researcher. And then there was this post back in Oxford to become a faculty member. That's when I came back, in year 2007. Time flies, so I have been here for another 14 years as, lecturer and associate professor. So I live with my husband and 14-year-old son. So hopefully I speak some of your language, because some of the language my son uses, I really don't understand. Have to say that. Okay, just to visualize where I have been to, okay? So I grew up in Wuhan, so the city I don't need to introduce, probably everybody now knows Wuhan. So that's where I was born, and until, I was 18, I lived in Wuhan. Then I chose to go to a city. Maybe I should get my point. Okay, I chose to go to a city called Dalian to do my undergraduate study, because one reason was that I wanted to be away from my parents as far as possible. The other is because it offered a special program where you can get 2 degrees. So basically we were taught chemical engineering in English. So that was the only this type of degree you can get in the country. And I was quite interested in chemistry, and I wanted to study abroad. So I thought that's a very good program for me. So it was a long journey, you can see. So I had to take a train, not the fast train you heard about in China nowadays. It's a slow train. So it's 2 trains, so I had to travel for 20 hours on the first train to Beijing, and then, transit there, and then another 20 hours on train to Dalian. So it, took me 3 days each way. So I made this trip 4 times a year to go there and then back in the holidays. So after that, then I came to the UK, so that's about 5,000 miles away from China. So now Oxford is my home, so with my son and my husband here. Okay, so that's where you see where I have been to. Okay, so now back to my research. So tissue engineering. Okay, so I think everybody maybe have thought about this immortality. So how we can live longer. So why we can't currently live to 150 years old. What's preventing us from there? So one of the things you may think about is in the old age our tissues or organs will fail. And so is it possible, like a car, where you can get some spare organs or tissues, so to replace the diseased ones or dysfunctional ones? So that's probably the start of, tissue engineering. So engineers and clinicians got together to think about how can we do that to grow some tissues or organs to replace the diseased ones. Okay, so because the recent advances in medicine has play wonderfully in stopping or slowing down many of the disease, but there are still things which we can't cure by taking pills or having surgery. such as, dementia, this Alzheimer's disease. And also there are things like if you have a kidney failure, then now you can have dialysis to keep you alive, but obviously your quality of your life has been significantly reduced. You have to go to hospital several times a week to do dialysis. So that's why in regenerative medicine, which includes stem cell therapy and tissue engineering. So this is what people envisioned to be able to provide a solution to this type of disease. Okay, so the gold standard when you have a failed organ is to have an organ transplantation, but we all understand that the demand always outstrip the availability, and this gap is growing bigger, as you can see here. Okay, so once we decide to do that, we need to look at what we are trying to make. So to look at the different tissues and organs in our body. So here I show some examples. So this is a picture of skin. So we all think skin is the simplest tissue in our body, but if you look at this, it's not that simple. It has different layers: the outside epidermis layer and then the underneath. So there are different types of cells involved. There are fibroblasts and also fat cells, nerve cells, and then there is this hair shaft, so hair, and also blood vessels. All this, this already looks quite complicated for the simplest organ. And this is a representation of liver, which shows these liver cells, they have to be arranged in a particularly way, how they can be next to each other, and the bile being collected in those bile ducts, and also the blood vessels. Okay, so they have to be, very well organized to be functional, and also they come in different, mechanical properties. With so this is a bone tissue, so it's mineralized, and the cells, the structure arranged in this concentrical units. So as you can see, our body, is a host of all these different tissues. So are we able to do any of this in the lab? Okay, so here I want to show you some of the examples tissue engineers has been able to achieve. So this is a picture of skin. So nowadays your doctor would be able to prescribe skin tissue, so which is not artificial material, but made of skin. So you can grow this, and then for say burn patients or people with like ulcers due to diabetes, your doctors will be able to prescribe this. Okay. And I don't know if any of you have seen this picture of a human ear growing on the back of a mouse. So this is of no clinical use, but it's just to show that we can achieve that, So this is where they make a biomaterial, a material of this human out ear shape, and then putting in some cells, so they would be able to grow this out ear of a human ear on the back of a mouse. And here is the story of where how engineers and clinicians work together to be able to produce this piece of trachea. Okay. So this is where they get donated, not donated from a dead person. They can get a trachea and then stripe off all the donor cells, then put in patient's own cells and grow them for a while and then transplant it. So this has saved a patient from collapsed lungs. So, you know, if they don't do this, they have to, so the patient wouldn't be able to breathe properly if with traditional surgery. So here I want to show you a video, okay, to see if, okay.
If you change your screen, I don't think I can see it.
Oh, sorry. Oh, okay. Yeah. Sorry, I think I forgot to, yeah, stop sharing this one and switch to the other one. Yeah, sorry, I will reshare this, I think, yeah. Okay, let's.
I think there's no sound, but I can see it.
Sorry, can you hear the sound?
No, I can't.
Oh, okay, I see. No, I'm sorry, but I hope, yeah, I think I forgot to, you know, switch this on. Yeah, and then.
Yeah, that's okay.
This is the first time that the whole cellular heart with blood vessel is printed. And another major breakthrough is that it is printed from the materials of the patient itself. The next stage is to mature these hearts in the lab for at least a month to help the cells or teach them how to interact with each other and how to provide electrical signal and to have a synchronous contraction or pumping ability of the heart. In a year or two, we hope to take these hearts and transplant them in rats or rabbits.
Okay. Yeah, just. Can you see it now? Yeah, back to the slide. Okay, so yeah, that's just some of the examples. So what tissue engineers has been able to achieve. Okay, so how do we do it? So it's very simple. Even Hollywood knows how to do it. So it's cells plus matrix. Okay, so basically, if I explain it in a simple term, is well, you have a scaffold which is of biomaterial. So whatever, you know, cells likes or of the property of your tissue. Okay, I just need to maybe get the point. Okay. And then you can make that into the shape of the tissue you want. For example, if the patient needs a tissue of this shape and size, then you make your biomaterials into this, and then you can add the cells and the growth factors. So growth factors are some of the molecules which can tell cells what to do. Yeah, if they need to grow or need to move around, if they need to become other types of cells. So these are the signaling molecules. And then we can culture them, grow them in a bioreactor with controlled environment. Okay, so over time, this will grow into the desired tissue and then be able to be transplanted for the patient. So this is the whole idea of tissue engineering. So in principle, we need the cells, we need the materials, we need this signaling molecules and the bioreactors. Okay, so you can see for those issues I just mentioned, yeah, so we need a large team of scientists to work on cells and bioreactor design, also to scale up, as you are not only making one, you need to make 1,000 so that they can be ready to be supplied to patients. And also the issue of preservation. So if you are to supply these tissues as a product, so you need to make them and then preserve them. So when it's demanded, when the patient needs it, you can ship it. Yeah, and also you need to think about if this is one product for all, you need to think about this immunocompatibility of your product. So how you can make it so the cells from say one donor would be acceptable for many different patients. And also there are material scientists working on biomaterials to make sure the materials, whatever you put in, is biodegradable. So over time, the material will degrade, but the cells will secrete their own supporting matrix to replace those. So in the end, the patient would be left with, you know, with the cells plus cell secrete the proteins as supporting material rather than some artificial materials. So this is, you know, what you need to make a tissue engineered product. Okay. Yeah. So as for the cells, so you must have heard about stem cells, yeah, which, you know, nowadays people have placed great hope on using stem cells as a, you know, therapeutic reagent. But I want to, you know, explain a bit more about different types of stem cells. So there is this human embryonic stem cells. So these are the cells of one a fertilized egg. They will divide to become these totipotent stem cells. And within, you know, a few days' time, it will form this blastocyst structure. Okay, and in here there are a group of cells containing inside this hollow sphere. So these are the embryonic stem cells. Okay, so scientists was able to, you know, from those in vitro fertilized egg which are donated for research, they are able to get those cells and then culture them in the lab. So these are the embryonic stem cells. And then these stem cells would be able to become all the different types of cells in our body. Yeah, so this essentially become the baby if they are left in the womb. Okay, and then these cells also can become other, we call adult stem cells. So because these embryonic stem cells are pre-fetus, yeah, but in our body we still have some stem cells which are very useful. For example, hematopoietic stem cells. These are blood-making cells. So when you heard about those bone marrow transplantation, actually these are the cells which play important roles there. So you transplant some bone marrow which has hematopoietic stem cells in it, so they can be used to making new blood cells for the patient. Okay, also there are neuron stem cells which you can find in the brain in some parts of our body. Also mesenchymal stem cells. These are the cells which again also in bone marrow, but they can make like muscle, bone, cartilage, you know, all the skeletal muscle systems. Okay, so there are different types of stem cells. So the embryonic stem cells and also the adult stem cells, which we have in our body. So, you know, the controversial which you heard about stem cells are mostly associated with embryonic stem cells, because this is where you have to destroy an embryo to be able to get these cells. Okay, so that's the different stem cell. So I don't know if you heard about this project called the London Project, which is how clinicians now can use embryonic stem cell derived cells to treat muscular degeneration. So this 80-something-year-old man, after, you know, being transplanted those stem cells, he gained some of the eyesight. So he is now able to read the newspapers, etc. So really, this is type of disease which we can't treat with traditional method. That's where we have to develop these cellular therapies for this type of disease. Okay, as I said, there are some other type of stem cells you can find in our body. Also, there are, maybe you have heard about T cell therapy. So T cell is a type of blood cell. They are not stem cells, but as they are immune cells, so this has been used by scientists. They try to, you know, help them to be able to treat cancer. So they found that if you can modify these cells in a way, they would be able to, you know, attack cancerous cells in your body. So that's another type of cell therapy, which, you know, scientists are currently working on. Okay, so that's sounds more like a clinician's work. And so how, you know, as an engineer, what we can do here in this field. Okay, so as I talked about, there are the many issues, you know, in tissue engineering, etc. So as you know, so one thing, you know, you can do is we need lots of cells. So those stem cells, it's you know, not only embryonic stem cells, but also the adult stem cells found in our body. They only come in very small numbers. So for them to be able to be used as a therapeutic reagent, we need the dosage, the necessary dosage. So we need to be able to, you know, scale up, so grow them in numbers, and also use automation, so that to make sure every time we got the cells of same quality, you know, not only quantity but also quality. So that's what engineers are good at. And also need to, you know, design the bioreactors to control the culture environment, okay? And so optimize the culture conditions so the cells are at their best environment, and also monitor what's happening inside. Because when we culture these cells, they need to be sterilized, so we can't constantly go in and take a sample or have a look. Yeah, ideally they are in this enclosed environment. So we need to design ways to be able to monitor how cells have grown. So are they healthy? You know, how many of them are there? Do we need to add more nutrients, etc.? Yeah, so that's how we need to monitor them. Also, as I said, we need to store these cells. So currently we cryopreserve them, means we store them at ultra low temperatures. But that's, you know, has some difficulty if it's a big chunk of tissue. For cells, it's mostly okay. So how we can store a tissue which we have grown there's also some challenges. Okay, so this is where, as an engineer, so for me as a chemical engineer, there are lots of things, you know, what I do, you know, as a chemical engineer, that's how I come into this field. So I just show you one example of the issues I work on in this particular field. So currently we can only grow very thin tissue like skin, yeah, this is like paper thin, or you know, a vascular tissue such as cartilage. So cartilage doesn't have any vascular, you know, blood vessel in it. So why is that? Okay, so how can we grow this, you know, a big 3D bulky tissue? So that's still currently a bottleneck in tissue engineering. So the problem is due to this blood supply. Okay, so if you think of any, you know, tissues in our body, you have this capillary network to supply nutrients to cells within it. So if you don't have this structure in place, you can imagine the cells, you know, within deep inside the structure would die quickly. Yeah, because if there are lots of cells and the nutrients can only be supplied from the outside, and you rely on diffusion to supply nutrients, then as the cell numbers grow, then the cells in the middle would die of lack of nutrients. Okay, so for the biologists, what they do is they can induce this angiogenesis, means blood vessel formation. Okay, they can add some molecules, so which would help blood vessel to grow into a tissue. But the problem is many of these molecules are associated with cancer, because you know, for tumor cells, they are very good at generating new blood vessels. So this is where those you know, factors or molecules come from. Okay, so that's not ideal. Yeah, if you want to put in some of the molecules produced by tumor into your body, that's you know, doesn't sound very good. So as a chemical engineer, so I have designed this bioreactor called the hollow fiber membrane bioreactor. So this shows how it works. So this, you know, thin these are the fibers. So the nutrient can flow in through these fibers. So these fibers, they have porous walls. So which means some of the molecules in the nutrients can diffuse out through the walls to feed the cells surrounding it. Okay, so this is more like creating an artificial network, capillary network. So the nutrient would be able to diffuse out to feed the cells. In the meanwhile, the cell metabolic waste can also diffuse in to be brought away. So by putting in these hollow fiber membranes, we are creating the capillary, you know, this artificial one, to help cells to survive. So this would means that we are not limited by the size of the tissue we can grow. Okay, so here are some of the prototypes we have made in the lab. So here is, you know, so we are having this tissue engineering solution for human problems. Okay, so I want to, also talk about some of the other projects I have done. So not only we make tissue for transplantation, but also we make those tissue as, for scientists to study. So nowadays, say if you want to study human brain, it's very hard to get a sample, yeah, as you can imagine. So you can study animal brain where you get, these brain slices, etc. But you can imagine nobody would want to donate that for you to study. So you either get diseased ones or you get, brain from dead people, etc. So it's this would damage the research, yeah, how you can get a material to study brain. So here we are trying to grow a piece of brain, which not for transplantation, but just for scientists to be able to understand it better. But it's not easy. As you can see, if we enlarge a section of cortex here, you can see here there are different layers with different types of neurons in it, and they are connected in a very structured way. Okay, so biologist was able to partially recreate this by, growing these cells in the neurosphere. So this means they throw in a bunch of cells and then they self-organize into this layered structure. But this is, again, has the problem as what I said, the size is limited and it's quite random. So you don't know if, you may get it this time, but not next time, etc. So we want to use the same concept as what I described to be able to grow a very structured brain tissue. So this is the concept where we will be able to. So this is the network we are going to make, and then we can use light to stimulate some of the cells and use some of the electrodes to detect the output. So in this way, to understand how cells connect to each other. And this is the like the design. So we have some light sensitive cells on the top, so they are able to be the receiver of the structure. And then we have this membrane so that we separate this group of cells from the neural network we created. And at the bottom, we have this multi-electrode array where we can detect the output. Okay, so we first need to get the cells. So here are some of the stem cell derived human neurons. You can see they are quite active. Yeah, they are trying to connect, so explore their surroundings and to see, which cells they can connect to. And if you stain them, it can show you different types of cells in the brain and also the, very extensive connections between each other. So we are able to recreate some of that in a 3D. So this just shows the network we have created with the help of hydrogel. So because brain is very soft material, so we use hydrogel as the matrix to support these cells. So you can see we are able to direct the this connection between the cells, so they are not random. We can organize them. Okay, so just compare this to what the biologist can do. So we partially achieved what we are trying to do, but we are not there yet to recreate cortex. So, but we can do with the engineering approach. Okay. So another very different project. So this is very recent. So through when I had a coffee with one of the previous students in the department. So after PhD, he went on to work for a consultant company. So he came back to say that, he saw, lots of issues with meat production, etc. As we all probably understand it now, that with the growing population and also with, even per person, we are consuming more and more meat, considering, many of the people in developing countries, as the living standard increase, they are consuming more meat. So which means we need to produce more meat, but with the current production already, so 30% of the surface is already used for livestock, and 18% of the greenhouse emission comes from this farming. Okay, so we are very limited on how much we can increase. Is there any way, we would be able to have alternative meat? Okay. So this is the company. So I then co-founded with this graduate of the department, so-called IV Farm Technologies. So this is to apply what I have described, the technologies used for tissue engineering. So that's for growing human cells as a therapeutic reagent. But here we apply that to animal cells, so growing lots of them, so growing meat like a plant. So these are the two sausages you see here we have produced in the company. Okay, so this is a spin-off company which have licensed some of the technologies I created in the university, so including the bioreactor and also the biomaterials, so to apply that to growing animal cells. So with a very recent study, it's very promising that cultured meat can have, have reduction in water usage, land usage, and greenhouse gas emission. Okay, so that's, so we can do that. Obviously, we are not the first one to do that. So in 2013, this Dutch professor, Mark Post, he has shown that he can create, grow this beef burger, with, you can see this large number of cell culture plates. He was able to produce this beef burger, but the cost is of $300,000. So who can afford this burger? Anyway, so yeah, there are more companies doing, different things like salmon, chicken, beef, etc. So we can all do it. And the cost has been significantly lowered to this $23 per chicken nuggets. Yeah, it's already significantly lower than 300,000, but still, would you routinely, have a chicken nugget for $23 each? Okay, so now there is still this scale-up challenge. If we put this into numbers, you will see the challenge. Okay. So there are approximately 100 billion cells in a kilogram of meat, and the world meat consumption is estimated to be 300 billion kilograms. So if we only to replace 10% of, that farmed meat with cultured meat, so this means we are to produce 30 billion kilograms of meat per year. And if you put this together, that means 3 times 10 to the 21 cells per year. Okay, if we are to have all these cells grow in bioreactors, This means we need to have 200,000 of these 10,000 liter bioreactors. So this number is, unbelievably large number, which we don't even currently have. So this is, well, we have this technology we normally use, in the medicine, but the volume we need is what we have seen in chemical industry. Yeah, this is the reactors you can see in the chemical industry, and in between this is currently people have in the pharmaceutical company. They have, cells or biomolecules produced. So there is still this scale-up challenge, how we can go even from here. That's what currently we can do to here in terms of culturing enough number of cells. So I would encourage you to think about this problem and hope you will help me to solve it, the future engineers. Okay, by that, I conclude. So thank you for listening and wish you all the success with your studies. And I'm happy to take any questions.
Thank you. Yeah, that was super interesting to hear about your work. We've got a couple of questions in the chat, so I'll just read them out and I'll start from the first one that came in. So the first one was, did you have to apply for grants to do research in Oxford?
Yes, of course. Everything I have to constantly apply to grants, funding. So this comes from, government funding source, or can be from collaboration with the industry. And sometimes the funding also comes from European Union. Yeah, so. Yeah, with Brexit, things becoming more challenging. But yes, that's one of our main tasks.
Yeah, I can imagine it becoming a bit more difficult. Next question is, how do you think stem cell therapy is going to evolve in the next 10 years?
I think it will be. I mean, when we started, for example, in the start of this century, everybody was optimistic, thinking, oh, in like 10 years' time, we will have a beating heart produced in the lab, etc. But then, yeah, of course, we still haven't had that yet. So I mean, with, now we started to see, as I have shown you, this stem cell therapy really have come into, clinical use in treating like a blindness and several different diseases. I think we are at a stage where we will see more and more stem cell therapy coming through, clinical trial and become available to patients. So I'm quite optimistic now. But for making tissues, that's, still lots of challenges remain. So I don't know if we will be able to produce a beating heart in the next 10, 20 years yet.
Yeah, I mean, like you showed in the picture, even the skin is so complex, and let alone trying to print a heart, that just feels really fast. But.
Yeah, and with the skin product I showed you, so that's doesn't even include all the components you have in your natural skin. So, it doesn't have hair follicles, it doesn't have nerves, it doesn't have, this, the color of your skin, those cells, which will, so they're still, but it can play the essential part of, covering up, etc. So yeah, there's lots to be done.
Yeah, great. So someone's asked, does using the material basis for the tissue directly from the patient completely eliminate the prospect of rejection?
Yes, depending on how you treat it, yeah, because the tissue is still from another person, and so there is a balance of how you treat this piece of tissue. So you want to, yeah, remove the cells, but if you treat it too harshly, you also remove some of the useful, proteins and molecules in that piece of tissue. So yeah, there's we call this decellularized matrix. So to remove the cells, but still, you need to optimize the process, making sure you get rid of all the materials which may elicit immunoreaction, but you want to retain all the useful stuff there.
Great, yeah, thank you. So do you think in future tissue regeneration technology could replace a large portion of a decaying brain to help with things such as memory in dementia patients?
Yeah, I think the hope now is more with stem cells. It would be able to regenerate rather than you implant a pre-made brain, like what I showed in a dish, and then put in a patient's brain. But it's more, you can put in cells and, some signaling molecules. So these cells would be able to do what they asked to do, to, make useful cells, make the right connections, etc. I think that's probably the treatment.
Great, yeah. And someone's asked if you've published any books or articles on stem cells, and they were asking if there was anywhere they could read specifically more about the bioreactors.
Yes, there are. I have publications, but they are very, technical. And but there are, if you search, there are. So even NHS provide lots of, basic knowledge on stem cells. With bioreactors, I did have, some books. I don't know how I can recommend. There are some, but mostly because it's very specialized research area, it's very technical. But if you even search in Google, you will find some of the articles which are more for general audience.
Yeah, great. So on a different topic from your work, someone's asked, have you faced any challenges in your career from being a woman specifically?
Not something I can, think of, but I think when I first, when I came back to this department as faculty, at that time we only have, I don't know, like a handful, 5 or 6 female academics. And we all working in biomedical area. So it was a very strange experience, but I can see now, in the department, we have more female academics, and also we have, lots of female students like you, and like research students. So I have many female DPhil PhD students in my group. So I think there has been lots of, development in female students taking up engineer as their subject and career.
Yeah, definitely. I feel like I was slightly worried about that before joining, like if there was just going to be way more males in the cohort. And I think the year that I started, it was maybe like 27% female, something like that. But I feel like the numbers are improving, and yeah. So someone's asked, are the bioreactors efficient in terms of energy, and how much pollution comes from bioreactors compared to the farming industry?
Because we haven't, had the final this scaled up production yet, so it's hard to compare because we are still trying to get there. But in general, as the study showed, as in one of my slides, yeah, they are, very efficient in the reduction in water, land, and the greenhouse emission. So, yeah, I think that we still need to produce, data from our own study, but that's what from other people's study that the data have shown.
Great, yeah. So we've only got 5 minutes left. So as one of our final questions, would you be able to share some tips on for anyone who's interested in studying or applying for engineering now?
I only have one piece of advice, is to follow your interest, because you don't know what this will take you to. Like myself, I never imagined I would be, doing this cultured meat or neural tissue engineering when I was in my high school. So I was just interested in chemistry, so I went on to read for chemical engineering, etc. So you never know where your final, career would be. Just follow your interest. That's my advice.
Yeah, that's great. I think that for me, that's what I was like specifically attracted to the Oxford or just general engineering courses in general, was you get exposure to a lot of things, and then you can actually find what you're interested in by actually doing it rather than trying to be told about it. Because the best way to know if you enjoy something is to go out and do it yourself.
Yeah, I think that's right, because when you are, 16, 18, you may not realize where your true interests are. So yeah, do whatever you like to do, you're interested to do, then you will find out later. Yeah,
great. Okay, I think we're closing up now. So there's just as a final note, there's another event tomorrow. If you want to head to that link, and you can find out some more details that you might be interested in. Thank you so much for coming, and thank you to Cathy for sharing about her work and answering some questions. And if you have any more questions, I'm sure you can just send an email to the outreach email, or if you visit the website, we also have a ambassador platform where you can send messages to student ambassadors directly, and we'll be able to chat with you about that through there. So yeah, thank you for coming, and hope you have a great weekend.